Extraction of RNA from virus. Mix all the reagents and gently invert 3 to 5 times before use. Add a 200 microliter sample to a 1.5 milliliter tube. Add 20 microliters of protease K. Add 200 microliters of lysis buffer. Add 300 microliters of isopropanol. Vortex for 5 seconds and mix at room temperature for 10 minutes with a thermomixer set at 1200 RPM. Transfer to a viral spin column in a collection tube. Centrifuge at 12,000 RPM for 1 minute and discard the flow-through. Add 500 microliters of washing buffer 1, centrifuge at 12,000 RPM for 1 minute, discard the liquid. Add 500 microliters washing buffer 2, centrifuge at 12,000 RPM for 1 minute, discard the liquid. Centrifuge at 12,000 RPM for 2 minutes. Leave at room temperature for 2 minutes. Add 40 to 100 microliters RNase free water to the center of the viral spin column membrane. Keep at room temperature for 2 minutes. Centrifuge at 12,000 RPM for 1 minute. RNA solution is ready for use or can be stored at -80 degrees Celsius. ERT-PCR Operation Part 1. The color and size of the centrifuge tubes used are specially prepared for filming only. Please refer to the instruction manual for actual specifications. Primer and probe dilution. First, dilute the primers to the concentration indicated in the instructions. Before using, flick each reagent tube to make sure the reagent in the tube is homogenized. Master mix preparation. The diluted primers and other reagents are pre-mixed in a centrifuge tube. Shake the tube lightly or pulse centrifuge the tube to pool the solution. Aliquot and label. Divide the pre-mixed reaction mix into clean centrifuge tubes of a suitable size. Label the tubes with the sample number or control type. Add negative control, sample, and positive control in sequence. When adding the sample or control groups, be sure to add group by group in the suggested sequence to the corresponding tubes. Ensure that each tube has been tightly capped before proceeding to the next group. Place the test tube in the centrifuge for short centrifugation to ensure that all reagents are pooled at the bottom of the centrifuge tube. Place the sample reagent mixture into a thermal cycler. Set the amplification profile according to the instructions. ERT-PCR Operation Part 2. The color and size of the centrifuge tubes used are specially prepared for filming only. Primer and probe dilution. First, dilute the primers to the concentration indicated in the instructions. It is recommended to turn off the lights when processing the fluorescent probe. During dilution of the fluorescent probe and the subsequent steps, avoid contact with light as much as possible. This demonstration video has an on-site light source for better shooting and display effects. The diluted primers, probes, and other reagents are premixed in a centrifuge tube. Divide the pre-mixed solution equally into PCR tubes. Add the products amplified in Part 1. The position of each sample should be determined before addition. When adding the samples and controls, avoid hovering the pipette over the PCR tubes. After each sample is added, it is recommended to seal the corresponding tubes tightly before adding the next sample. Water can be added into the empty tubes to avoid deformation during the heating stage. After all samples are added, make sure that all tubes are tightly sealed. If the tubes need labeling, avoid the transparent part in the center of the tube cover so as not to affect the fluorescent reading. Place the tube rack into the centrifuge for a short centrifugation to ensure that all reagents are pooled at the bottom of each centrifuge tube. Mix the reaction by this scraping action. Place the sample reaction mixture into a real-time quantitative PCR machine, qPCR machine, and set the amplification profile according to the instructions. Confirm that the machine successfully receives the correct amplification profile information. Experimental precautions. There is a risk of contamination in the gene amplification area, so a special amplification area or a separate special laboratory should be set up for amplification only. There must be a strict physical separation between nucleic acid extraction and reagent preparation areas. Spray the biosafety cabinet with 10% of bleach before the experiment. Then turn on the UV lamp and irradiate for 30 minutes. Materials such as pipette tip boxes, plates, and centrifuge tubes are sprayed with 10% bleach before being placed onto the test bench. Prepare the reaction mixtures in the reagent preparation area, mix well, then aliquot and centrifuge. Transfer of the prepared reaction mixture to the designated laboratory for sample addition is performed by designated personnel or through a transfer window. In the sample addition biosafety cabinet, add the nucleic acid sample into the corresponding reaction tube and tightly cap each reaction tube. During the mixture preparation or sample dilution, mix thoroughly by up-down actions of the pipette to avoid vibration and aerosol formation. It is recommended to change pipette tips frequently. Avoid error in volume and cross-contamination caused by repeated use. A sample table should be made to record the position of added samples. Special care should be taken to tightly cap the PCR tubes. New gloves are recommended. After reverse transcription PCR, the tubes are taken out and centrifuged. After the experiment, disinfect the laboratory tabletops and floor with effective chlorine disinfectant or alcohol. Turn on the ultraviolet lamp for more than 30 minutes for disinfection. All novel coronavirus-related medical wastes need to be sterilized with high temperature and high pressure, and then treated as ordinary medical wastes.